Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. Sexual reproduction in flowering plants with artificial pollen germination and hybridization. If you like this video, subscribe my channel and click on bell icon to get notifications. Like, follow and share my videos to your students and friends to get more information in biology related to NEET and other national state level competitive exams. In this video, I am going to explain artificial pollination process along with artificial hybridization in plant breeding programs. Pollen germination is a process. In this, the development of pollen tube it takes place from germpore of exine. It is called pollen germination. This pollen germination process it occur naturally when the pollen grain it reach the compatible stigma. During this process, the pollen grain or microspore, when it reaches the stigmatic surface, now it contains two cells. In this, one is vegetative cell or pollen tube cell and another one is generative cell. Now the pollen tube cell or vegetative cell, it grow into a pollen tube through germpore. This pollen tube helps to travel generative cell. During pollen germination, the stigmatic surface it provides all suitable conditions for the development of pollen tube. This pollen germination is a process generally it is affected by various environment factors. In this, the pollen germinability and viability, longevity, it play an important role in pollen germination. The external factors like temperature, humidity, light, it affects the process of pollen germination generally. Along with that, some of the internal factors like food materials present in cytoplasm in the form of stored food, along with its quality and number of enzymes present in cytoplasm of pollen grain, it affects pollen germination process. During pollen germination, the pollen grain it need some suitable conditions. Let us discuss what are the suitable conditions for pollen germination. During pollen germination process, the stigmatic surface it play an important role during pollen germination by secreting various secretions. In these secretions, it contains lipids, gums, sugars along with resins as a main components. One of the element it is boron. This boron it play an important role in pollen germination by stimulating pollen tube growth. Pollen viability is a very crucial step. It helps in artificial pollination and artificial pollen germination process. It is different in different species and different genera. Pollen grains are generally having a stain that express orange or bright red color generally. To observe the pollen grains with a staining procedure, in this procedure we have to take 1 gram of potassium iodate and 0.5 grams of iodine, or both are dissolved in 100 ml of distilled water. After dissolving these in distilled water, now it is called IKI solution. The pollen viability counts vary made within 5 minutes after pollen it was placed in IKI solution. The knowledge of these surrounding conditions and suitable conditions for pollen germination it helps in artificial pollination and artificial pollen germination program. Artificial pollen germination It takes place in laboratory. Now such pollen germination is called in vitro pollen germination. In vitro pollen germination, the culture medium is a technique that stimulates the conditions of a style stigma, inducing germination and pollen tube growth. The requirements for artificial pollen germination. In this, the first requirement for this, it is a high relative humidity, suitable temperature. These are useful as a physical factors. Some of the chemical compounds like carbohydrates are needed along with some elements like boron and calcium. Along with this, some of the enzymes are rarely used in artificial pollen germination. These are cellulase, pectinase, calase enzymes. Along with these, we use some hormones like IAA and GA, indole acetic acid and gibberellic acid as a hormones. Artificial pollen germination process, it is conducted in laboratory with the help of a method. It is called hanging drop method. In this method, 10% sucrose solution, it is used as a artificial synthetic medium for pollen germination. Along with sucrose solution, we use boric acid, calcium ions, magnesium ions, potassium ions to get better result. For this experiment, we have to collect the pollen grains from different flowers like pea flower, chickpea flower, crotalaria, blossom and vinca rosea flower pollen grains. After collecting these pollen grains, the pollen grains are transferred in a drop of sugar solution or sucrose solution. In hanging drop method, first we have to prepare a nutrient solution by dissolving 10 grams of sucrose in along with 10 milligrams of boric acid in 100 ml of distilled water. Now it becomes 10% sucrose solution. By taking a clean slide, we have to drop a few drops of sucrose solution on this clear slide. After that, the pollen grains are dusted on this slide, especially in sucrose solution which are collected from a suitable flower. After completion of this process, we have to observe this slide under microscope after few minutes. Especially after 15 to 30 minutes, by observing this slide under low power lens microscope, we can found that some of the pollen grains can produce pollen tube as a outgrowth that indicates pollen germination. In this slide, we can calculate the percentage of pollen germination by using a formula, that is percentage of pollen germination is equal to small n by capital N into 100. In this formula, a small capital N, it denotes that total number of pollen grains observed under microscopic field. Small n, it denotes the number of pollen grains germinated under same microscopic field. By using these values, we can calculate the percentage of pollen germination in laboratory. After that, the next topic is artificial hybridization program. In artificial hybridization program, generally by conducting a cross in between two different plants of same species, we can produce some of the hybrid varieties in agriculture field to enhance food production. Hybridization is a process. In this, crossing of two genetically different individual to result in a third individual with a different, often preferred a set of traits which are not seen in parental generation. In artificial hybridization process, in which only desired pollen grains, desired means the plant which have superior characters, such plants are considered as desired plants, their characters are called desired characters. Such type of desired pollen grains are used for pollination and along with fertilization process. Pollen pistil compatibility, it is essential and it is useful for successful pollination and fertilization process. Once compatible pollen is accepted by the pistil, immediately the next events like fertilization take place, whereas if the stigma is incompatible, in such condition the transferred pollen grains are rejected by the stigma in hybridization program. Artificial hybridization, it is a process. Generally, it is achieved with the help of various steps. In this, the first step is emasculation. The second step is bagging. The third step is cross-pollination. And fourth step is rebagging. In this, the first step, it is emasculation. Emasculation is a process. In this, generally, the anthers are removed from bisexual female parental plant. In this, without affecting female reproductive organ, it is referred as emasculation. For this emasculation process, first we have to take a floral bud, and this floral bud generally it is opened with the help of forceps. After opening this flower or floral bud, the stamens are in immature condition. That means these are not matured at young stage. In this immature condition, these stamens are removed with the help of this forceps from floral bud. After removing these anthers, now in that floral bud, immature pistil present. That is called emasculation. The main importance of this emasculation is this emasculation process generally it prevents self-pollination. One more important thing, the emasculation is no need to perform in case of unisexual flowers, because unisexual flowers having either stamens or pistil both are not present in unisexual flower. If the unisexual flowers are used in artificial hybridization program, in such condition no need to perform this emasculation. The second step is bagging. Now the floral bud after its maturation, there is no chance for self-pollination, but sometimes if the flower is exposed to surrounding environment, in such condition the stigma it may receive the pollen grains of other plant. To avoid such condition, we perform bagging. In bagging, it is a technique used to prevent fertilization of stigma from undesired or another plant pollen. For this, the emasculated flower it is covered with a polythene bag. It is made up of butter paper. This butter paper can allow gases exchange. This process is called bagging. The main intention of this bagging is to avoid contamination or pollination with another flower. The third step is cross-pollination. When the emasculated flower it having pistil, when the pistil is in matured condition, in such condition the pollen grains are collected from desired plant with the help of brush or needle. Now by collecting this pollen grain, the pollen grains are transferred on selected emasculated flower stigmatic surface by removing bag. The fourth step is rebagging. After completion of this pollination, this pollinated flower again some chances are there to receive the pollen grains of another flower, so that To avoid such type of contamination, this pollinated flower, it is again covered with same polythene bag. It is called rebagging. Artificial plant hybridization, it helps to produce hybrid plants with desired characters. These hybrid plants are generally used in agriculture field to enhance food production. Thank you for watching. If you like this video, subscribe my channel, like and share my videos to your students and friends.